All right, Nima. So please
Okay.
go ahead.
Okay. Hello, everyone. My name is Nima Barani. I'm a computer science master student at the Memorial University of Newfoundland. And today I want to show you my research project, which is the prediction of host pathogen RNA interaction from RNA sequencing and dual RNA sequencing data using variation autoencoders and supervised machine learning methods. And during the course of bacteria infection, bacteria and host cells negotiate for their survival. Understanding the interaction between host cells and bacteria pathogen is essential for development and improving of therapeutics. And as far as we know, for the first time in this study, we want to implement a supervised machine learning model to predict activated genes during an infection using dual RNA sequencing data and variational autoencoder. Like every machine learning model problem, we need data. And to obtain the data, we need to convert the raw sequence reads to which are the strings that consist of ACTG characters into count metrics through a bioinformatic pipeline. And the image here actually demonstrates our count metrics, and the rows here are gene IDs, which are our features, and the columns here are our samples, which correspond to different conditions of an experiment. And let's take an experiment that we infect a host cell. And for example, at the beginning of the experiment, I mean time 0, we take a sample, and that would be our first column. And then after 2 hours, we take another sample, and that would be the second column, and so on. And each number here represents to the expression level of each gene that is here. But having a high or low level of expression doesn't necessarily mean that a specific gene is differentially expressed, and statistical analysis tests are needed to identify such genes. And by no different tests like F-test, the Wald test, the likelihood ratio test were performed to identify the differentially expressed genes. But the problem with this approach is that the statistical tests are computationally intensive, and as a study in 2020 was performed, they demonstrate that by increasing the number of cells in single-cell RNA sequencing datasets, the time is increasing. And for example, in a dataset with 10,000 cells, which is a common number in bioinformatic datasets, it may take up to 50 minutes to perform the test. And also, it requires a lot of memory computer for performing the test. So with the advent of machine learning, the machine learning algorithms improve the computing time, the storage requirements, and the memory utilization in analysis of RNA sequencing datasets. But before we feed our data into machine learning models, we need to know that RNA sequencing data is considered as a high dimensional data, and it suffers from the curse of dimensionality problem, as the number of features which share our genes are way more than samples. And to tackle this problem, we have to use dimensionality reduction algorithms. Previously, algorithms, the famous ones, for example, PCA and TSNE, were used to convert data from a higher dimension to a lower dimension. But PCA is mainly related to dimensionality reductions, linear dimensionality reductions, and also it considers that our data has a normal distribution. And TSNE is also mainly used for data visualization tests. Several bioinformatics studies have applied the single-cell RNA sequencing data and input it into variation autoencoders, and they successfully demonstrated that variation autoencoders has a superior performance compared to algorithms like PCA, ICA, UMAP, and others. And they also showed that variation autoencoders generates features that retain biological patterns and leads to more accurate classifiers. And what is the variational autoencoder? Variational autoencoders are like autoencoders. They are neural networks composed of an encoder and a decoder. And they are trying to minimize the reconstruction error between the encoded decoder data and the original data. But the main problem with autoencoder is overfitting. So in order to introduce some regularization to latent space, there's a slight modification to autoencoders, and that is instead of encoding an input as a single point, we encode it as a distribution over the latent space. And this is the workflow for our project. And by now we already have collected our data through a comparative literature review, and then we already performed the data preparation task, which was the converting raw sequence reads to count metrics, and it's mainly a bioinformatic task. And by now we have to implement the variational autoencoders and other dimensionality reduction algorithms, and implement our machine learning models, which are classification models like random forest, SVM, decision trees, and multi-layer perceptrons, and compare the performance of each algorithm with each other, actually, and finally choose the best combination. I mean, the combination of dimensionality reduction algorithm and the machine learning model, and tune the parameters and hyperparameters and make it reproducible. And this is my references. Thanks for your attention.
All right, Nima, thank you very much.
You're welcome.
Questions from the audience?
Hi, Nima.
Hello.
Hi, thank you for your presentation. I have one question. I saw that your related work, they all have different data sets, especially the two related work at the end, they were doing the same task. Are you going to use the same data set as they use, or is there any benchmark for comparing related work, state of art?
Okay, sure. Actually, this data is single-cell RNA sequencing data, which is different from my data set, which is dual RNA sequencing data. And yeah, and it's completely different. And the benchmark, actually, they use different performance metrics, and yeah, that's it.
So which is the data set you're using?
Actually, I collected my data set through literature review in bioinformatics. After, in each publication, the authors usually put the accession code to the data set, and I'm going to use them, and they are dual RNA sequencing data sets.
Okay, so you're using more than one then. Okay.
Yeah, exactly. Yeah.
Thank
Multiple. You. Yeah, you're welcome.
All right. Thank you very much.